Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing TIFR Biology question paper conducted in the year 2011. If you are new to my channel, please do like, share and subscribe. Coming to the year 2011. First question. A couple wants to have two children who are genetically identical. Short of having identical twins engineered, which of the following statements best describes the number of offspring they would have to allow this? So they should have at least a billion offsprings to get to this condition where the children are genetically identical. So answer is option D. The next one. I have a sequence of DNA that is 24 base pair long. Starts with a start codon and ends with a stop codon. That codes for the protein called MFP. Which of the following statements about the amino acid composition of the protein are false? They have asked for the false statement. Adding a 25th base pair has no effect. The DNA sequence is only 24 base pair long, so even after that means, and it includes the start codon as well as the stop codon. So after the adding 25th base pair will not have effect. That is correct. Substituting the 6th base may have effect. Yes, it can result in change in the amino acid sequence. Then adding a base after the 6th base and deletion of the 10th base has a smaller effect than each mutation alone. If you add a base after the 6th base and you delete a 10th base, there is going to be a frame shift mutation and it will have larger effect, not smaller effect. So that is the false statement. So answer here is option C. The next one. An alcohol molecule with a 5 carbon ring can be chemically modified at positions 3, 4, 5 only to generate biologically active second messengers. The modification can occur either at a single position or in more than one position in combination. How many distinct second messenger species can be generated in this manner? So the modification can occur either at a single position or in more than one position or in combination also. So how many second messengers will be produced as a result of this? So it can be modified at position 3 or at 3 and 4 or at 3, 4 and 5 or at position 4 and 4 and 5 or at position 5 and 3 and 5. So totally there will be 7. Answer will be option C. Then I set up a cross between two genetically identical parents heterozygous for a recessive mutation that causes the progeny to die as embryos. Which of the following is false? So a cross has been set up between genetically identical parent heterozygous for a recessive mutation. That means capital A small a into capital A small a. That is the heterozygous condition, and for a recessive mutation. Small a, if you have obtained that in the progeny, they will be homozygous for the recessive condition. So recessive mutation, that progeny to die as embryos, and small a condition will cause death of the embryos. So which of the following is false? A quarter of the embryos show the mutant phenotype. Yes. Capital A, capital A. Capital A, small a, capital A, small a and small a, small a. Small a is shown by a quarter of the embryo. So that is correct. 3/4 of the adult progeny show no phenotype. That cannot be correct because 3/4 will have phenotype and they will be surviving the embryo stage. A third of the adults do not pass the mutation to the next generation. That is this capital A condition, they will not pass the mutation to the next generation. All the adults are phenotypically indistinguishable. That is also correct. All the adults can be distinguished phenotypically because they have different genotypes, capital A and capital A small a. So the answer here will be option B. A signaling pathway I have identified has three proteins A, B, C that work to allow function X. When A is absent, X does not happen. The absence of C or B individually does not substantially affect function X. When C and B are both defective, function X fails but can be restored if excess A is provided. Which of the following statement is false? Protein A functions after protein C and B. So protein A functions after protein C and B, that is correct because this is a pathway. So first here they have told that C is the absence of C or B. Absence of C or B individually does not substantially affect function X. If either C is absent or if B is absent, it is not going to affect function of X because C can give rise to A, B also can give rise to A, and A can allow the function X to happen. When C and B are both defective, function X fails but can be restored if excess A is provided. When C and B are both defective, if both C and B are defective, then A will not happen. So function X will fail. Now if you provide A from outside to this particular pathway, then function X will happen. So protein A functions after protein C and B, that is correct. Then protein A requires protein C and B. It doesn't require both the proteins. It requires either C or B. That is what they have told here. The absence of C or B individually does not substantially affect function X. So if C is absent and B is present, or if B is absent and C is present, A will be produced and you will get function X. So B is the wrong statement. Protein A requires protein C or B to function. Protein C and B do not influence each other's function. So the answer here is option B. When bacteria are transformed with DNA, the effectiveness with which they take up the DNA is quantified as the transformation efficiency. If the operational definition of transformation efficiency is number of bacterial colonies obtained when bacteria are incubated with 1 microgram of DNA. So that is the definition of transformation efficiency. That is number of bacterial colonies obtained when bacteria are incubated with 1 microgram of DNA. A new student prepared bacterial cells suitable for transformation. On testing his preparation, he obtained 8,000 colonies when he incubated the cells with 8 picograms of DNA. His transformation efficiency is. So they have already mentioned the formula for calculating transformation efficiency, that is number of colonies obtained when incubated with 1 microgram of DNA. So number of colonies divided by 1 microgram DNA. So 8,000 divided by 8 picograms. Convert picograms to micrograms. So 8 into 10 raised to minus 6 microgram and you will get the answer 1 into 10 raised to 9. Answer is option A. Then next one. At the plasma membrane of a cell, the transduction of light is mediated by the proteins A, B and C in the sensor and A, B and C. A is the sensor and C is the effector. The efficiency of this sensory transduction system is limited by the probability of interaction of A with B and B with C. If the probability of A interacting with B is 0.2 and the probability of B interacting with C is 0.1, what is the probability of the signal passing from A to C? So the answer here will be multiply both these values. 0.2 into 0.1 you will get 0.02. Then next one. A species of bacteria found at the bottom of a shallow pond has a hard cell wall and a semi-permeable membrane. Which of the following statement is true? The cell wall prevents the cell from being crushed by external forces. The cell wall prevents the cell from exploding due to internal pressure. The internal and external pressure are equal, hence the cell wall determines the shape. Answer is option C. Then next one. Pigment color in guppies is determined genetically. Of the progeny obtained from a guppy pair, 25% exhibit red pigment, 25% exhibit yellow pigment, 25% white and 25% exhibit orange pigment. Based on these observations, which of the following statement is correct? So they must be codominant alleles because they are equally expressed colors. So red and yellow are codominant alleles. Answer is option B. The next one. An accidental exposure of mouse embryo to the UV light results in a green pigment patch in the skin. However, none of the F1 and F2 progeny exhibit green patches in the skin. The most probable explanation for this phenomenon is the mutation is repaired in the germ cells. Now, if the mutation was present in the germ cells, then it would have expressed in the F1 and F2 progeny. Now, the mutation is repaired in the germ cells means it is not going to be expressed in the progeny. So, that could be the answer, one answer. Then, this is a somatic mutation and does not get transmitted to next generation. Now the mutation is happening in the somatic cells. So naturally it will not get transmitted to next generation. Only if it is the mutation happening in the germ cells, then it will be transmitted to the next generation. But it is somatic mutation, so it is not transmitted to next generation. So it does not be visible in the F1 and F2 progeny. So B is also correct. The statement gene is imprinted and hence is not expressed in the progeny. That is also correct. It will be inherited either from the mother or it can be inherited from the father. So, and it will not be expressed in the progeny. That is also correct. So, all of the above are correct. The next one. A species of flightless bird found on an island is 10 times bigger than its nearest known mainland relatives. This could be because predators are absent on the island. Answer is option C. The next one. Injection of an extract from lotus leaves in a rabbit induces immune response. The characterization reveals that some of the antibodies can selectively recognize a protozoan antigen. Also, the infection assay revealed that the rabbit become immune to protozoan infection. So here you extract something from the lotus leaves and you inject into rabbit, it induces an immune response. Now this immune response, what you do is you isolate the antibodies and characterize them and you find that these antibodies can recognize protozoan antigen. So naturally the rabbit will become immune to the protozoan infection because it produces antibodies against protozoan antigen. So based on this observation, it is claimed. So they have claimed something that lotus leaves have an alkaloid that induces production of antiparasitic antibodies. Which of the following observation will weaken this claim substantially? The pond water from where lotus leaves were taken show very high parasitic count. Now that is not going to weaken the claim because very high parasitic count in the pond water means it doesn't have anything to do with the rabbit immune response. You only take the extract from the lotus leaf and inject into rabbit. You are not taking the pond water and injecting into rabbit. So it is not going to weaken the claim substantially. Lotus leaves do not have alkaloids that have been previously reported to have antiparasitic activity. That is also not correct. That will also not weaken the claim. When the lotus leaf samples from 50 different ponds were screened, only 10% showed increased antibody production when injected in rabbits. Now that is a very low number. The only 10% means only very few uh, lotus leaf extract showed production of antibodies in the rabbit. So that will weaken the claim substantially, because when you are taking samples from 50 different ponds, as per the claim, what they have mentioned in the question, you should have got at least around 80% or more than 80% um, increased antibody production when injected in rabbits, but you got only for 10%. So that will weaken the claim substantially. So answer is option C. Then next one. Tight junctions are apically placed junctions in the epithelial cells. These junctions are selectively permeable and regulate the water and electrolyte transport across the epithelium. The transepithelial electrical resistance measurement reflects how permeable an epithelium is. So TER, it will reflect how permeable the epithelium is, and TER is inversely correlated with epithelial permeability. So lower TER values indicate leakiness. Lower TER value means higher will be the epithelial permeability and it will result in leakiness of the tight junctions. It is known that one of the cholera toxin called SOT increases the leakiness of the tight junctions, contributing to symptoms of cholera. A pharmaceutical company is screening for bacterial toxin which can enhance the epithelial permeability. Their aim is to use the best of these toxins as an agent to enhance the transport of anti-cancer drug across the intestinal epithelium. So the pharmaceutical company is screening for bacterial toxins that will enhance the epithelial permeability. So they have to enhance the epithelial permeability means the TER values must be low. And what is their aim for? The aim of this pharmaceutical company, that is they want to transport anti-cancer drug across the intestinal epithelium. It is known that transport of the drug increases when TER is below 5000 ohms. The effect of these four toxins and toxin plus drug, that is figure B, on TER were tested on cultured epithelial cells. The treatment started after 3 hours and the toxins and drugs were washed out after 4 hours of incubation. Now this graph I have made it clear in the next slide. So this is the graph, and what they have asked is which of these would you recommend for use in combination with drug for clinical trials. Now if you see figure B, this is toxin D. Toxin D is having a lower TR value, lower than 5000. Similarly, in figure A also toxin D is having lower TR values. So that is below 5000. So in this case, answer will be toxin D. Then next one. A graduate student is attempting to purify an enzyme from the brain of a blowfly. He grinds up the head of 500 blowflies in a buffer solution and filters to obtain a crude extract. He then employs two-step purification to end up with a moderately pure protein. He estimates enzyme activity at each stage of purification. So what does the student do? He wants to purify an enzyme from brain of blowfly. So he grinds around 500 blowflies in a buffer solution and he obtains the crude extract. Then he purifies the crude extract using two-step purification procedure and he ends up with a moderately pure protein. He estimates enzyme activity at each stage. So specific enzyme activity and total enzyme activity has been assessed. Now questions. Which of the following statements about step 1 of purification is true? The data reflects the normally expected pattern of a protein purification project. Now there is if the data reflects normally expected pattern of a protein purification project. For a normally expected pattern of a protein purification project, there should be decrease in total enzyme activity and increase in specific activity for normal purification table. But here if you see, after the step 1 of purification, the total enzyme activity has risen drastically. Then in step 2 it has decreased. So it is not a normally expected pattern of a protein purification project. Then B, the increase in total enzyme activity is unexpected. That's correct. I have explained it now. The decrease in total protein is unexpected. Protein will decrease. The increase in specific enzyme activity is unexpected. Enzyme activity will increase. Specific enzyme activity will increase. Then next one. Which of the following statements about the data are plausible? The enzyme being purified constitutes a large fraction of the total extracted protein. No. Only a small fraction of the enzyme was recovered in this project. It is not very small. The tissue contains a substance that inhibits the activity of the enzyme. That is correct. Okay, the tissue contains a substance that inhibits the activity of the enzyme. That is why you got first around total enzyme activity as 500 and then it increased. That means that inhibitory compound was removed after the first purification step. That is why enzyme activity increased. So the tissue contains a substance that inhibits the activity of the enzyme is correct. Not only that, if you see specific activity also increased drastically after step 2 of the purification. So all these will show that the tissue contains a substance that inhibits the activity of the enzyme.